Sorry, bear with me here. All right, audio check. Audio check. That's the right microphone. AI generate. Oh, switch to live captions. All right, audio check. Okay, sorry about that. I just have to double check things. Okay, so tonight we are getting deep into chapter 13, which is, if you remember, on the cytoskeleton and cell movement. And then we'll get into a little bit on chapter 14, which is the plasma membrane. And then we'll finish up with the beginning of chapter 16, which is an introduction to cell signaling. So when we left off, we were talking about the actin molecule. And now in section 13, point 2, we're going to talk about the myosin protein. Actin and myosin are two very prevalent proteins in the cell, very important components of cytoskeleton and cell movement. So in this specific lecture, we're going to look at how myosin proteins use ATP hydrolysis to generate mechanical force, and they do that along the actin filaments. Myosins are among the best studied molecular motors, and they serve as very good prototypes for understanding how chemical energy is converted into movement. From things like muscle contraction to transport of organelles and substances inside of the cell, structures inside of the cell. So, if you look at this topic, it really bridges things like structure and bioenergetics and cell physiology and helps us understand how cells achieve motion at both a microscopic and a macroscopic level. All right, so this slide introduces myosin. It's the prototype of all ATP-driven molecular motors. So if you remember back from A&P, if you take an A&P or simple, the muscular system, if you covered it in Bio 2, muscle fibers, right, which make up muscles, are large multinucleated cells. And they're multinucleated because they form by the fusion of myoblasts. And then within each fiber, you have these cylindrical bundles that are called myofibrils. These myofibrils consist of actin, which we call thin filaments because they look thin under the microscope, and myosin, which we call thick filaments because they look thick under the microscope. And they're arranged into these repeating contractile units, which we call the sarcomere. Right? The sarcomere is bounded by two perpendicular lines, which we call Z-discs. So how the sarcomere shortens is explained by what's called the sliding filament model. Right? And basically, what happens is myosin heads, which is a particular domain of the protein, literally walk along actin filaments, and they pull them inward and subsequently shorten the sarcomere. So, it's this very organized alignment of actin and myosin that gives skeletal muscle both its striated appearance, right? Striated meaning it's filled with striations or lines, and also it gives the skeletal muscle its mechanical strength. All right, so here you can see this figure shows a very detailed structure of a muscle fiber. Sorry. You can see that the skeletal muscle tissue is composed of bundles of long multinucleated cells, which we call muscle fibers. Each of those, remember, is formed by cell fusion, which is why they're multinucleated. Within each muscle fiber are many myofibrils, and each myofibril is divided into these repeating units called sarcomeres. Those are delineated by Z-discs. So the alternating arrangement of the thick myosin filaments and thin actin filaments creates those light and dark banding patterns, which are very characteristic of striated muscle. So this figure, gives you a visual foundation so that we can understand how the molecular interactions now translate into tissue level contraction. All right, so here we're elaborating on the sliding filament model. All right, so during contraction, actin and myosin filaments slide past one another. The actin filaments move inward towards what's called the M line. This shortens the sarcomere, but it does not change the length of the filaments. The filament length remains unchanged. The Z discs are just basically dislocating, right? They're changing location. The major motor that drives this process is the protein myosin 2. This is a very large, actin-based protein that has 2 heavy chains, each containing a globular head and a long alpha helical tail, and 2 pairs of light chains that are both essential and also regulatory. And the tails coil together to form dimers, which assemble into thick filaments. Each myosin head binds both actin and ATP. And that's important because it's coupling ATP hydrolysis to force generation. All right, so here in this figure, we see the organization of filaments in a sarcomere and the structure of the myosin 2 molecule there on the right. All right, so you can see the electron micrograph shows the repeating sarcomeric pattern where the thin actin filaments anchor at the Z discs and the thicker myosin filaments are centered at the M line. So this shows how actin slides towards the center during contraction, shortening the sarcomere. That's why we say the distance between the Z discs decreases. But these filaments do not change length. All right, this second figure shows myosin 2. You've got 2 heavy chains with globular head regions and long tails, and they coil together in a dimer. The head domains are the motor sites. These are the regions that hydrolyze ATP and interact over and over again with actin to drive contraction. All right, so a little more on muscle contraction. So, thick and thin filaments, how they're arranged and stabilized within the sarcomere. When you look at the thick filaments, they consist of hundreds of myosin 2 dimers, and they're arranged in a staggered array. The thin filaments made of actin anchored at Z discs, and then these other important proteins. So nebulin aligns the actin filaments to maintain a uniform length. Remember I said they don't change length. So nebulin kind of gives it some additional integrity so that it doesn't change length. And also tropomyosin, another important protein, is important in regulation as it regulates myosin's access to actin binding sites. So think of like tropomyosin is sitting there blocking. It blocks and prevents the myosin head from latching onto the actin. Right? The orientation of filaments are reversed at the M line, and that's what produces the symmetrical contraction. There's another protein called titin, and titin is actually, if I remember correctly, it is the biggest protein in human cells. It's very big, elastic protein, and that's an important property of it. It goes from the Z discs to the M line, and it acts as a molecular spring that keeps thick filaments centered, and it restores the sarcomere's resting length after contraction. All right, here you can see we can reinforce what we just talked about. Thick myosin filaments, they're composed of many, many, many myosin 2 molecules, and you can see them interacting with actin filaments that are anchored to the Z discs. So we call this connection here, we call it a cross bridge when it's binding to the actin binding sites, and that's occurring during contraction. The proteins nebulin and tropomyosin and titin, these are considered stabilizing proteins, and you can see them in this figure as well. And tropomyosin is a protein that can block the myosin head from binding to actin. Nebulin is a protein that gives this some additional rigidity. And then titin is a very elastic protein you can see runs all the way through the myosin. And when this thing, after it's done contracting, it's that elasticity from the titin protein that allows the Z disc to go farther apart again. Okay. Also, you see the polarity of actin and myosin. It reverses at the M line. All right. And that's important so that contraction pulls equally from both sides towards the center. And that ensures a very uniform shortening throughout the sarcomere. Right. So this is the structure and the function of the sarcomere. And now we're going to get into a little more of the mechanism and signaling. So the cross-bridge cycle, I think ATPase cycle of myosin is what describes how ATP hydrolysis powers this mechanical motion. So where it starts, the cycle begins when ATP binds to myosin. The binding of ATP causes it to detach from actin. Now, next, hydrolysis of ATP, in other words, hydrolysis of ATP to ADP and inorganic phosphate, it cocks the myosin head, which is in a state of potential energy. So this energized head then binds to a new site on actin. The phosphate releases, and that triggers the power stroke. And this is where the heads pivot and pull the actin filament toward the sarcomere center. Finally, ADP release resets the head for then another cycle. And these will cycle like a lot and cycle quickly, as you can imagine, which is required for a muscle contraction. And each cycle consumes one ATP molecule, and it moves actin about 10 nanometers. But it's the repetitive, coordinated activity among millions of heads contacting millions of actin filaments that truly generates the macroscopic force of muscle contraction. And additionally, calcium is very important. We're going to talk about that here in an upcoming slide, the role of calcium in the signaling. So, but this figure here goes through the cycle that we just talked about, the cross-bridge cycle. You can see there at the top, first ATP binding dissociates myosin from the actin. Then ATP hydrolysis changes the conformation of the myosin head, and it moves it into a cocked, highly potential energy position. The head can then rebind actin at a new site further along the filament. Inorganic phosphate gets released, and then the power stroke is performed. Right? So that's the motion that physically moves the actin. Finally, ADP is released, and a new ATP binds, restarting the cycle. Right? And it's cycling this thing from bottom to top over and over again. The binding, hydrolysis, conformational change that results in very directed movement. All right, so now, how do you get from, innervations, nervous system innervation into, to the cell and then resulting in the system of contraction? Well, there is a signaling event, and that signaling event is regulated by calcium. So muscle contraction is regulated by calcium, is regulated by calcium ions. The thing about calcium is its signaling mechanism is fast. I mean, it'll quickly diffuse in solution, and it can quickly get pumped out of the cytoplasm of the cell, where it's stored in a region of the cell called the sarcoplasmic reticulum. So the sarcoplasmic reticulum is a calcium sequestering component of the endoplasmic reticulum, right? So in a resting muscle, the protein we talked about, tropomyosin, which runs along the actin filament, in a resting muscle, it's blocking the myosin binding sites, right? So even if the ATP cycle is going and myosin 2 head is trying to access actin, it can't. Tropomyosin, imagine it's like a rope that wraps around actin and prevents the binding of myosin 2 during a resting state. All right, so there's an additional protein complex that can interact with tropomyosin called the troponin complex. The troponin complex is composed of troponin 1, okay, troponin C, and troponin T. Right, so these three proteins, which make up the troponin complex, control the blockade, right, of tropomyosin blocking the attachment of the myosin 2 head to actin. All right, so you can see the roles of these different proteins. TN1, it's what actually prevents the actin-myosin interaction. TNT, it anchors this complex to tropomyosin, so it is there to work. And then finally, the third protein, TNC, is what binds to calcium that comes out of the sarcoplasmic reticulum. Okay, all right, so when calcium is released from the sarcoplasmic reticulum, it binds to TNC. That causes a conformational shift that moves tropomyosin away from the binding sites. This ultimately allows myosin heads to attach to actin and initiate cross-bridge cycling. Right? When the stimulation ends, calcium is quickly actively pumped back into the sarcoplasmic reticulum by calcium pumps that are on the membrane. And then tropomyosin recovers the sites, right, where actin will bind, and contraction stops. Right? So this is a quickly and highly reversible system that really ensures very precision timing between, as a bridge between electrical excitation from the nervous system or innervation and mechanical force resulting from the cross-bridge cycle that follows after calcium signaling. Okay, so here's actin. Now, tropomyosin is not shown in this figure, but imagine it's trying to get, somewhere there. It's got a figure labeled in the figure, myosin binding site blocked. So tropomyosin is like a rope that wraps around actin and prevents myosin 2 from sticking. Here's the troponin complex that is attached to tropomyosin, right? It was the TNT protein. And it's the TN1, the blue, that's blocking the site. Okay. What was I talking about there? So, so when calcium gets released from the sarcoplasmic reticulum, it binds to TNC and it causes a conformational change. And now you can see the actin, the actin myosin binding sites that are exposed. And now myosin 2 can bind. Okay. So, this troponin complex is part of the on-off switch for contraction. Right? You can see the cross-sectional view that shows how the exposure enables actin-myosin interactions to proceed. Very elegant system and also extremely well understood. All of these proteins have been crystallized. It's really, this is one of the mechanisms of the human physiology that we really know everything about. It's pretty remarkable. Okay, moving on a little bit. That is contraction in non-muscle cells, but myosin 2 is not only important in skeletal muscle. It has roles as a molecular motor in non-muscle cells as well. Right? And it can generate localized contractions with the cytoskeleton. So in non-muscle cells, what happens is these like bipolar filaments of myosin 2, they basically slide actin filaments in opposite directions to produce tension. So, tension is important in a cell because it gives rigidity and it gives it shape and structure. And that's why we call these contractile assemblies being found in something like a stress fiber, right? So, stress fibers basically anchor to focal adhesions, which we talked about in the last section. The other component that has these bipolar myosin 2 filaments sliding past actin filaments are adhesion belts. Adhesion belts are important in linking epithelial cells together. And then, of course, the contractile ring, which wraps around the cell during cytokinesis and pinches it into two to divide the cytoplasm after mitosis. All right, so two figures here. This one showing myosin 2 function in non-muscle cells, right? So this is what I mean by a bipolar myosin 2 filament, right? So see how they're linked up like that, and it's pulling actin filaments in opposite directions, which is unlike what happens with skeletal muscle, right? Actin sliding in the same direction, and that gives tension, right? Just like two people that are stuck together pulling on a rope in opposite direction. All right, this is a fluorescent image showing the contractile ring during cytokinesis and how that operates. That should be familiar to many of you, to result in daughter cells following mitosis. All right, so let's see here. Yes, so also in non-muscle cells, myosin 2 activity is controlled by phosphorylation rather than the troponin-tropomyosin system that's found in skeletal muscle. So when intracellular calcium goes up, there's a protein, a signaling molecule called calmodulin. And this is a very important regulatory protein and calcium sensor. And when calcium binds to it, it forms a complex that activates myosin light chain kinase. Okay, myosin light chain kinase, or MLCK, is a protein that phosphorylates the regulatory light chain of myosin 2. That causes a conformational change that unfolds the molecule and promotes filament assembly. So this phosphorylation activates myosin's ATPase activity, enabling it to bind actin and generate tension. Then when calcium levels fall, there's an enzyme called myosin light chain phosphatase that removes the phosphate and returns myosin to an inactive state. Okay, so here you can see calmodulin, the important regulatory calcium binding protein. And this is really considered a signaling pathway. It's a very short, simple one. I guess this would be not the first one we learned about, but this is important event in calcium signaling. So it's some people call it the calcium calmodulin MLCK pathway, but here calcium is binding to calmodulin, and that resulting complex activates MLCK. MLCK is a kinase, which means it phosphorylates, and it phosphorylates the regulatory light chain of myosin 2. And this modification converts myosin from a folded inactive state into an extended filament-forming conformation. And this figure really doesn't show that or demonstrate that very well, because you can see it's kind of look the same. But, it's just becoming, it makes it into an active state. Right? But when it's activated myosin can now interact with actin to generate contraction. So in this instance, reversible phosphorylation acts as a molecular switch. Right? These are for non-muscle cells, enabling cells to rapidly regulate contractile activity in the cell. All right, so we've talked about skeletal or muscle myosin. We've talked about non-muscle myosin, and now we're going to talk about what are known as unconventional myosins. So not all myosins form thick filaments or drive contraction. We have what are called unconventional myosins. These include myosin 1 and myosin 5. So just pop quiz, which myosin have we learned about today? We learned about myosin 2, right? Myosin 1 and myosin 5 can function as cargo transport motors that move along actin filaments, and they move towards the plus end. Right? Myosin 1, for example, That's a single-headed. It's single-headed. It has a short tail and can attach to vesicles or membranes. It uses ATP, and it literally moves along actin tracks. Myosin 5, on the other hand, a little different. It's got two heads, and it's got this long coil-to-coil structure. So we haven't really talked much about coiled coils. They're a relatively uncommon protein secondary structure. The secondary structures that are most ubiquitous within the cell are certainly the alpha helix and the beta sheet, but we see these in these motor proteins, these coiled coil structures. Anyways, so myosin 5 has two heads and a long coiled tail, and the tail will move, right? So it basically takes multiple steps without detaching it. And this myosin 5 is important. It can transport organelles, vesicles, and it can even transport intermediate filaments all around the cell. And it's that we're not going to talk about intermediate filaments. We're going to skip that section in this chapter. But these unconventional myosins really highlight like the versatility of the myosin family of proteins and what it does beyond muscle contraction. Okay, so you can see these unconventional myosins working. Myosin 1 over here, right? It's myosin, and it's moving a vesicle along actin. See how it does that? It kind of attaches and it pushes that way. So single-headed motor. That's the big difference here. Myosin 1 is a single-headed motor, and it's moving towards the plus end. Okay, and it's carrying membrane-bound cargo like vesicles, right? And that's because it lacks the long tail that's needed for like filament formation. It's myosin 1 does not generate contraction, but instead it mediates membrane trafficking. All right, in this we have myosin V, much bigger myosin protein than myosin 1. It's a two-headed motor, and it literally walks along actin filaments, and it can transport big things, large organelles and intermediate filaments throughout the cell and move them around. So what's really cool about this section 13.2 is we really focus a lot on the chemistry and cell biology, the biochemistry. It's an introduction to molecular structures and function, but the stuff is moving. Stuff is it's living, right? It's alive. So that is it for myosin. We're going to move on to an even weirder cellular protein, which is called the microtubule. Okay, so we're going to look at, in this section, the structure, the assembly, and how microtubules, one thing that characterizes them is how dynamic their assembly and disassembly is. And that, these microtubules serve as like the cell's internal highways. They serve as scaffolds. They're essential for intracellular transport, the position of organelles, the movement of chromosomes during mitosis, and it even has an important role of determining the shape of cells. So they don't work alone. Microtubules work in concert with motor proteins, right, like myosin, and also other components of the cytoskeleton. Helps organize the cell's interior and orchestrate movement. Okay. All right. So we've talked about actin, we've talked about myosin, and now we're talking about tubulin. Those are the three main, protein families for these cytoskeletal and motor proteins. So microtubules are cylindrical. Imagine a pipe, like a PVC pipe. That's what they're shaped like. Just, a little bit on that. Why would they be shaped in the shape of a pipe as opposed to making something solid? Well, it's more efficient. It takes less mass to build a circular structure. And it's also practically just as strong. And, because it takes less mass, it can assemble and disassemble more quickly, which is really a very important component of microtubules. Now, things do not travel inside the tubule, as far as I don't think it's ever characterized that. But because they can so rapidly grow and shrink, um, it benefits them to be in the shape of a tube, of a tube. All right, so they're cylindrical polymers, and they're built from alpha and beta tubulin dimers. Okay, they're arranged head to tail, and they're arranged head to tail into 13 protofilaments that form this hollow tube. So, what is a protofilament? Well, if a microtubule is like a hollow metal pipe, then think of the protofilaments as like individual metal strips that wrap around in like a helical kind of shape to create the pipe. All right, so each tubulin dimer binds GTP. Right? GTP is it interacts with tubulin very rapidly, high affinity. So, and it's very important that GTP hydrolysis is very important in the polymerization of these microtubules. So each tubulin dimer binds GTP, but only the GTP on beta tubulin is exchangeable and regulates the polymerization. This arrangement gives the microtubule inherent polarity, gives it a plus end, which can grow very quickly, and it gives it a minus end, which grows very slowly. Or the minus end could just be anchored to a structure, such as the microtubule organizing center, abbreviated the MTOC, important structure in the cell. All right, so a little bit on the polymerization, like the tubulin dimers, they add preferentially to the plus end, and they add to it already bound to GTP. So the tubulin dimer has GTP on it, and it readily binds to the plus end. When it starts getting added on to the plus end, the structure that gets built is like a piece of paper. It's flat, like a sheet, and then it rolls up into the cylindrical tube. Right? Once it's incorporated into the microtubule, the GTP on beta tubulin hydrolyzes to GDP, and that weakens the binding and predisposes the filament to depolymerize. Right? And it's this really brilliant architecture, think about it, that allows microtubules to be very stable and flexible and can support very rapid reorganization when needed. All right, so we got two figures here. Don't let this scare you here. It's a lot of pieces, but we'll go over it in detail. So these two figures are giving us a very visual understanding of how tubulin dimers assemble, right? And how they assemble into a functional microtubule. So here we've got the alpha and the beta tubulin dimer. And it's emphasizing that alpha tubulin permanently holds GTP, while beta tubulin has an exchangeable GTP or GDP. Okay? The microtubule is composed of 13 protofilaments. There they are right there. And they alternate alpha and beta, you can see. So they alternate alpha and beta tubulin, and they wrap around to form a microtubule cylinder. Okay. All right, so like this part here is here on this figure right here. And this is demonstrating the polymerization. All right, so, and it's demonstrating the role of GTP in polymerization. So tubulin dimers with GTP bound over here. So there's the dimer, and it's got GTP bound to it. The GTP is bound to the beta tubulin, and this adds to the plus end, right? The rapidly growing end. And you can see the structure that initially forms is like an open sheet. So this sheet then zips closed, and it forms a stable tube. And then shortly after assembly, you can see GTP is hydrolyzed into GDP, and that actually weakens interactions, between these dimers and within the whole lattice. and ultimately forms, the plus side. So that weaker orientation, I'm sorry, did I say plus? I meant minus side. That's what eventually forms the minus side. So on the minus end, dimers with GDP are more likely to dissociate from this end. This is a very like nucleotide dependent balance between growth and disassembly. And that is the most important component of microtubules. It's this dynamic ability to, rapidly grow and rapidly disassemble. Okay. So we got a term called dynamic instability that kind of describes that component of microtubules. So they exhibit this, and it means that they're constantly alternating between phases of growth and shrinkage. And this is, important in allowing the cytoskeleton to remodel very rapidly. The mechanism depends on the GTP cap at the plus end. Okay, so there's a GTP cap at the plus end. And as long as GTP bound dimers are added faster than the GTP is hydrolyzed, the cap stabilizes the growth filament. However, if hydrolysis catches up, the GTP cap is lost. Right? This loss leaves exposed GDP tubulin. That destabilizes the lattice, triggers rapid depolymerization, and they have a special name for this process. It's called catastrophe. Right? Now, catastrophe is not finite. Growth can resume if new GTP tubulin adds, and this is a rescue event. So what's happening in dynamic instability is basically a cycle between catastrophe and rescue. All right, the balance among polymerization, GTP hydrolysis, and rescue is what defines the organization of the microtubule network. All right, there are very well characterized compounds that destabilize microtubules. A very famous one is colchicine, which will cause mitotic arrest. Because it prevents, and it forms mitotic arrest at M phase, right? Because it prevents the microtubules from pulling the chromosomes apart. So it works by binding free tubulin dimers and preventing the addition to the tube. That ultimately collapses the microtubule system. There are also drugs that stabilize microtubules. The drug Taxol, right, which comes from, so Taxol is a chemotherapy compound that the last time I learned about it, it was extremely expensive because you have to it comes from a yew tree and it's in very small amounts of the yew tree. So that it locks microtubules in a polymerized state. You think about it, that also prevents mitosis, right? And since cancer cells are rapidly mitotic or highly mitotic, Taxol is an effective chemotherapy agent. Okay, so here you're seeing the process of dynamic instability in detail. On the left, we're looking at the growth, right? And on the right, we're looking at the shrinkage of a microtubule, okay? So here we've got a GTP cap, right? And over here, we're seeing the loss of the GTP cap, okay? So, and it's that basically which triggers shrinkage. I don't really particularly like this figure very much. I think it's overcomplicated, but you can see during growth, it's growing here at the plus end, GTP that's bound to these tubulin dimers, right? And it's producing a cap here, but if that cap is removed, it's destabilized. Okay. All right, so now that is, microtubules in a nutshell. However, it just doesn't magically decide to depolymerize or repolymerize or catastrophe or rescue. There is important proteins and enzymes that mediate that behavior of microtubules. So the behavior of microtubules is tightly controlled by a very important class of proteins called microtubule associated proteins. And we're not going to say that 100 times here on out. We're just going to call them MAPS. Right? Microtubule associated proteins are MAPS, and they regulate the growth, the shrinkage, the stability of microtubules. So they bind primarily to the plus end of microtubules and influence whether they elongate or shorten. All right, there's different types. You've got the polymerases. These accelerate growth by increasing the rate of the GTP tubulin addition, which sounds pretty generic, but that's basically what they do. You've got the depolymerases, which remove GTP tubulin subunits from the plus end, and that promotes catastrophe and eventually disassembly. And then finally, you have class proteins, which have a completely opposite effect by stabilizing the shrinking microtubule and facilitating rescue. But it's through a combined action of these MAPs that the cell can really tune the microtubule dynamics to match whatever physiological needs it has, right? Keeping the spindle stable during mitosis or allowing flexibility during migration, whatever is needed can be accomplished with these polymerases, depolymerases, and class proteins. And that's